C'est bon? Okay. So thank you for the kind introduction, and thank you everybody for being here today. And welcome to my to the defense of my PhD. So as Céline said, which was about the adaptation, structural diversity, and phylogenetic relevance of a novel functionalized domain containing membrane architecture in Archaea. So first, I would like to start by introducing quite briefly what Archaea are. So at first, we considered life to be divided into two main domains. On the one hand, prokaryotes, which are characterized by relatively small organisms, pretty simple and lacking nucleus. And on the other hand, the eukaryotes, which have much bigger cells, far more complex, and also have a nucleus. However, we also found some strange organisms that have, for instance, very specific metabolisms like methanogenesis, or inhabit very extreme environments such as hypersaline lakes, but also hydrothermal vents that didn't really fit with this quite simple dichotomy of life. So in 1990, Carl Wöse and colleagues introduced a third domain to group these very strange organisms that they called Archaeobacteria and later on Archaea. But due to the very small number of Archaea that we knew at the time and the very specific environments they inhabit, Archaea were at first considered only as very strange extremophilic bugs. But thanks to the advent of the genomic era that we currently know, the diversity of Archaea that we know just burst, as you can see with this phylogenetic tree of the diversity of Archaea. Of course, we could identify new phyla in extreme environments, but completely new diversity of Archaea was also found in more moderate environmental settings, such as open ocean, but also groundwaters, sediments, and lands and soil, but also associated with higher organisms. So now it is widely accepted that due to the significant part they represent of the biosphere, that Archaea play critical functions in all geochemical cycles on Earth. So Archaea are actually quite important for the biosphere. But they are quite also different from the others. While they have similar features than bacteria, such as their overall cell structure and also the organization of their genome, they also have features similar to those of eukaryotes, like how they process their genetic information. But in contrast, they also have very unique features, such as their lipids. So lipids are amphiphilic molecules that are composed of a hydrophilic region, the polar head, and a hydrophobic core, the hydrocarbon chains. As you can see here, the polar head and the hydrophobic core. And polar head groups are mainly conserved in between the three domains of life, but that's about all that is similar between archaeal lipids and the other types of lipids. Indeed, if we look at the bacterial and eukaryotic lipids, they are built upon straight alkyl chains here that are ester linked to a glycerol in a 1,2 configuration. While in Archaea, the hydrophobic core is constituted by a branch isoprenoid chain here that is ester linked to a glycerol in a 2,3 configuration. So all these characteristics have been associated with enhanced parameters of the membrane in Archaea, such as the compaction and the rigidity of the membrane. But Archaea are also able to synthesize very specific lipids that are bipolar. So they have a polar head group on each side of the molecule, and they are much bigger than the typical monopolar lipids in Archaea. And as such, these bipolar lipids, also called tetraeters, can form monolayer membranes that are even more stable, rigid, and impermeable than the typical bilayer of Archaea. But bacteria and eukaryotes are also able to synthesize lipids based on polyisoprenoids, just like Archaea, in the form of either opanoids for bacteria or sterols in eukaryotes. These two compounds have very interesting functions in the membrane of bacteria and eukaryotes, as they can modulate the physicochemical properties of this membrane. But Archaea lack these two compounds, but instead they can synthesize other polyisoprennoids that are this time straight chains that are also called acyclic polyisoprenoid hydrocarbon, or APH, that just like sterol and opanoids in bacteria and eukaryotes regulate the membrane properties in Archaea. So altogether, the specificity of the Archaea lipids enhance the stability of their membrane and were thus associated with their adaptation to extreme conditions. In bacteria and eukaryotes, all these lipids form bidimensional leaflets that are called membranes, and by gathering depending on their molecular affinity. So for instance, we can have regions that will be enriched in sterols or opanoid with saturated lipids, for instance in blue here, while in red here we have the unsaturated lipids that are pushed aside. So we can actually segregate different population of lipids in different regions of the membrane, which generate differential physicochemical properties of this domain in the membrane. And these different physicochemical properties also allow these different regions to insert or ensure different sets of proteins. So this allows to compartmentalize the membrane of bacteria and eukaryotes into different functionalized regions, which are extremely important for the membrane of microorganisms, as it transforms a relatively small and small membrane into a highly efficient and multifunctional platform. But due to the specificities of the Archaea lipids, we don't know how they organize into a functional membrane and whether this model for bacteria and eukaryotes can also be applied to Archaea. But one model has been drawn for Thermococcus bauophilus, which is a hypothermophilic and piezophilic archaean, which is able to synthesize these monopolar dieters in green, 65% of the membrane, 35% of tetraeters bipolar lipids in purple, with the addition of trace amounts of these acyclic polyisoprenoid hydrocarbons in black. Due to the very high proportion of dieters in the membrane of Thermococcus bauophilus, it is highly likely that at least part of the membrane of Thermococcus bauophilus is actually in the form of a bilayer, which stability is questionable under the extreme conditions Thermococcus bauophilus lives, even though it is based on Archaea lipids. But to explain the stability of these bilayer domains, our lab has suggested and later on experimentally demonstrated that this apolar polyisoprenoid here in black can insert in the middle, in the midplane of the bilayer, and thus increase its stability under extreme conditions. When we look at the 3D rendering of such a model membrane organization, what we can see is that we can actually segregate monolayer domains that are composed of our tetraether lipids in blue here. And on the other side, we have this apolar polyisoprenoid containing bilayer domains in red. So just like in bacteria and eukaryotes, we have these different populations and different regions of the membrane which have different physicochemical properties and thus can ensure different sets of proteins. So we have a model organization that is quite similar in the 3 domains of life and involves the segregation of the functions in different regions of the membrane. So the main objective of my PhD was to determine whether this organization was a specificity of Thermococcus bauophilus or if it could be applied to other Archaea. Unfortunately, while in bacteria and eukaryotes we have means to directly observe the structural organization of the membrane into different domains, for instance, using fluorescence microscopy, tagging either lipids or specific proteins, we don't have this means in Archaea, so we don't have any direct way to observe membrane organization in Archaea. But to circumvent this issue, this limitation, and look at our main objective, we used the lipid composition in order to infer the membrane organization in Thermococcus bauophilus and other Thermococcales. So we will embark today in a journey to the center of the membrane by first analyzing the outermost layer of the membrane, so the polar head groups and intact polar lipids. Then we will dig further into the hydrophobic region of the membrane by looking at the core lipids, so the lipids without their polar head groups. And finally, we will see the innermost layer of the archaeal membrane, where these apolar polyisoprenylide hydrocarbons are supposed to lie. As our goal was at first to determine whether our organization could be extended to other archaea, of course, we will look at this lipid composition in a variety of archaea and environmental settings. But before diving into my result, I just want to make a quick point about the methodologies that we used to analyze lipid. So first, we started from a cell pellet here that we grow in the conditions that we wanted to test. And the first step is to completely destroy the integrity of the cell and disrupt these cells in order to extract the lipids. So this can be done by several physical, chemical, or enzymatic methods. And once these cells are disrupted, we can extract the lipids using usually solvent mixtures that are composed of either of both a polar and a non-polar solvents in order to solubilize the two parts of the lipids, the polar head groups and the hydrophobic region. These extracted lipids are then separated using chromatography in order to get isolated populations of lipids that are much more easy to analyze and identify, notably using mass spectrometry. So our model organism was at first Thermococcus barophilus. As I said, it's a hyperthermophilic and piezophilic organism that has been isolated from the snake pit hydrothermal vent, a deep sea hydrothermal vent located in the mid-Atlantic ridge here, pointed by the stars. But we have a lot more of closely related archaea to Thermococcus barophilus, which are called the Thermococcales. I put here the phylogenetic tree of the 50 strain of Thermococcales that we know of. Here is Thermococcus barophilus, but we have isolated very different species that inhabit a wide range of hydrothermal systems that can be either terrestrial and freshwater springs or like Thermococcus barophilus, deep sea and marine systems. So the Thermococcales, due to their diversity in both genetics and environmental settings, they offer quite a good view of the diversity that we could see of membrane organization. So first, let's start with the identification of the polar head groups and the intact polar lipids. So there was only one characterization of the intact polar lipids of Thermococcus barophilus by Martenson and colleagues in 1999. And what they revealed is only one polar lipid, which was based on a phosphoinositol polar head group here, attached to a diater. But we know from the genome of Thermococcus barophilus that it has the capacity to synthesize a whole lot of other polar head groups, such as phosphoglycerol here, also phosphoserine. And in addition, other intact polar lipid composition has been reported for Thermococcales, and again, they showed other polar head groups than only phosphoinositol. For instance, if we take Thermococcus codacarensis here, we can see a whole lot of polar head groups, such as other phosphoexoses and different derivatives, like phospho-N-acetyl exosamine here, or phosphodiexoses with different derivatives again, and finally glycolipid like this dihexose here. We have also shown that only a fraction of the lipids from Thermococcales can be actually extracted and analyzed. So we hypothesized that most of the polar head groups of Thermococcus barophilus remain unknown, and that we have things to actually discover in this archaeon. In addition, most of the polar head groups that have been described for Thermococcales have been on dietary lipids in green here. But we know from the core lipids of this archaea that tetraeters, so the bipolar lipids in purple here, represent a significant portion of their membrane. But which polar head groups are actually attached to these lipids remain largely unknown, and even more than for dieters. But this was results that have been obtained more than 20 years ago. So let's see what the technical improvement that we have done can gives us in terms of intact polar lipid composition in Thermococcus bauophilus. So here you have a chromatogram of Thermococcus bauophilus intact polar lipid. The first thing we can see is that we have this major peak here, which corresponds to the typical phosphoinositol dieters that have already been reported for Thermococcus bauophilus. But we could also identify other polar head groups. For instance, 6 of them were previously reported in Thermococcales, but were for the first time in Thermococcus bauophilus, such as phosphoglucose, phospho-N-acetylglucosamine here, or different derivatives of phosphodiexiose like this one. In addition, we could also identify for the first time completely new polar head groups, such as other derivatives of phosphodiexiose bearing either 1 or 2 N-acetyl groups, like here, or acetidine diphosphate, like here. But altogether, when we look at the diversity that we could reveal for Thermococcus bauophilus, all of the polar head groups were actually based on hexoses. And again, we couldn't find any traces of the expected other polar head groups, such as phosphoethanolamines or glycolipids. In contrast, we could still observe intact polar lipids based on other polar core structures. Finally, we identified in Thermococcus bauophilus tetraeder-based intact polar lipids, such as this one, bearing 2 phosphoinositol head groups. But we could also report quite new core structures in this Archaea, such as an extended version of the typical dieters here, or a dieter bearing an additional methyl group here, but again with phosphoinositol polar head groups. And finally, we also detected a new tetraeder core structures, which is based, as you can see here, on a 3-alkyl structures, internal structures, but again bearing phosphoinositol polar head groups. In this table, I displayed the lipid composition of Thermococcus bauophilus and the proportion each lipid represents of its total lipid that we could identify. What we can see at first is that the typical PI dieters again represent a vast majority of Thermococcus bauophilus intact polar lipids. But more dramatically, the tetraeders that we identified here represented less than 1% of all the intact polar lipids that we could observe in Thermococcus bauophilus. To get a larger view of the polar head group diversity that we could see in Thermococcales, we did a similar analysis on 6 additional strains, which are here, such as Palaeococcus elgesoni, Pyrococcus furiosus here, or Thermococcus onurineus here. We could identify polar head groups in these strains that were quite similar to those of Thermococcus bauophilus, such as N-acetylglucosamine here, or tetraeders bearing these 2 phosphoinositol. However, we still had the same problem as PI data was always the most abundant IPL in all these 7 Thermococcales, as you can see here, even in species which we know synthesize a vast majority of tetraeder core lipids, such as Pyrococcus yayanosi here. So altogether, this result suggests that we might have some limitation in extracting and analyzing Thermococcales lipids, and we tried to estimate these limitations. So we quantified the lipids that we could detect in Thermococcus bauophilus, which amounted for only 0.1 femtogram of lipids per cells, distributed between 99% of dieters and 1% of tetraeders, and bearing only phosphoexoses as polar head groups. While we could identify a large diversity of new polar head groups, this is still far from what would be expected for Thermococcus bauophilus. For instance, up to 10 femtogram of lipids per cell are expected and distributed between 30% of dieters, 70% of tetraeders, and with additional polar head groups that we didn't see here. So this questions how representative the lipid that we described for Thermococcus bauophilus are of its complete membrane lipidome, and suggests that much effort are still required in order to grasp the entirety of its lipidome. To estimate how limited was our extraction procedure, we hydrolyzed our total lipid extract. So we removed the polar head groups from the lipids we injected in the machine to detect them. And as you can see, when we do that, we can reveal a much higher content of lipids, including more tetraeters. So this suggests the difference between the detected and extracted lipids, that we have some limitation, to identify and detect these lipids. So for instance, some of them could be quite reluctant to the ionization method that we use, and thus stay invisible to our mass spectrometer, or could just stay stuck on the column that we used and just never reach the mass spectrometer. We did the same thing to estimate the total lipid content of Thermococcus bauophilus by hydrolyzing directly its cells. And again, we could reveal a much higher quantity of lipids. And this time, the ratio between dieters and tetraeters was actually reversed. So again, the difference between total lipids and extracted lipids informed us on what limitation could be, how flawed was our extraction procedure. So first, what I suggest is that some of the lipids of Thermococcus bauophilus are just reluctant to the extraction that we used. So they are not soluble in the solvent that we used, for instance. Or that the lysis method that we employed was actually not able to disrupt all the cell of Thermococcus bauophilus. So altogether, this result clearly show that we have some limitation in analyzing intact polar lipids in Thermococcus bauophilus. So to circumvent these limitations, we shifted our analysis to core lipid in Thermococcus bauophilus. So we analyzed the lipid without their polar head groups again. The core lipid composition of Thermococcales has been largely described in Thermococcales, as you can see here in this table by Shugai et al. In 2004. However, they usually proceeded by the same intact polar lipid extraction, and then they removed the polar head groups to analyze the core lipids. So their analysis is should be biased as ours was. To exemplify this, we conducted the same analysis of hydrolyzing the intact polar lipids from Pyococcus furiosus, as you can see here. This revealed the typical core lipids that we identified in Thermococcus bauophilus, such as these green dieters here, but also the D-alkyl tetraeters in purple and the tri-alkyl tetraeters in blue. But when we hydrolyzed directly the cell pellet of Pyococcus furiosus, we could actually reveal a lot more of different core structures, as you can see here. These new core structures that we identified were mostly unsaturation isomers of the typical tetraeters, and this unsaturation isomers contains cyclopentan rings, which are the equivalent of the unsaturation in bacterial lipids, from 1 to 4 for these D-alkyl tetraeters, and from 1 to 2 for these tri-alkyl tetraeters. And finally, here we could identify a completely new core structures for Pyococcus furiosus, which we will call monoalkyl tetraeters, as it has an internal covalent bond in between the two alkyl chains. And again, this new core structures was discovered with different unsaturation isomers. So we have quite a lot more of core structures in Thermococcales when we directly hydrolyzed their cell pellets. So we did the same thing for the 53 strains of Thermococcales that we had in the lab. So here you have, again, the phylogenetic tree of Thermococcales and their lipid compositions here. The color code is the same as before, dieters in green and tetraeters in purple, blue, or red. The first thing that we could observe is that all our strains of Thermococcales could synthesize both dieters and tetraeters. And as the foundation of our model organization for the membrane of Thermococcus bauophilus, this suggests that they could potentially organize their membrane in the same way. As all the thermococcales possess these three types of lipids, this also suggests that it could be an ancestral features that have been inherited from their common ancestor. But as you can see here, the thermococcales exhibit quite a large variety of lipid composition that we try to assess here using the proportion of dieters in red. So as you can see, this proportion range from 8% in Pyrococcus auricochi here to 80% in Thermococcus coalescens. So we try to sort this variety out by drawing statistically supported groups of Archaea depending on their lipid composition. We came out with three groups. In green, we have the species that produce more dieters than tetraeters. In purple, those that produce more tetraeters than dieters. And finally, in red, those that produce large amounts of this monoalkyl red tetraeters. When we superimpose these groups on the phylogenetic tree of the thermococcales here, well, we can see that the colors are quite randomly distributed in the tree, which suggests that there is no phylogenetic signal or no clear phylogenetic signal in the lipid composition of these strains. And so closely related strain of thermococcales don't necessarily have similar lipid composition. So if the evolution and the specific evolution of these strains couldn't explain the lipid composition of thermococcales, we hypothesized that it could come from the environment they live in and the environmental conditions we grew them. So we try to correlate these conditions with the lipid composition of our strains, as you can see here. The first thing we can see here is that the optimal temperature negatively correlates with dieters and positively with tetraeters, which actually suggests that species living in higher temperature settings will tend to have more tetraeters than those that live in a lower temperature environment, which is something that has been already observed in Archaea. But we could report for the first time a new correlation between the optimal salinity and the types of tetraeters these thermococcales produce. And for instance, species living in higher salinity will tend to have more dialkyl tetraeters and less monoalkyl tetraeters than those that live in lower salinity environment. Interestingly, we didn't see any correlation with the optimal pH of these strains. And while this trialkyl tetraether was detected in every strain of thermococcales, it did not correlate with any of the environmental parameters that we tested here. So its adaptive function remain unknown. So now we have a good understanding on how dieter and tetraeters distribute among thermococcales. But now let's see the distribution of these apolar polyisoprenoids in black. So thanks to the work of a fantastic PhD student of our lab, Marta Salvador Castel, we now know that apolar polyisoprenoids insert in the midplane of the membrane, so here. And doing so, they induce lateral structuration of the archaeal membrane and also increase the membrane stability under extreme condition. So this suggests that this apolar lipid could actually support very interesting adaptive functions in Archaea. However, the data that we have on these compounds is very limited. For instance, we only know the apolar lipid composition of two thermococcale strains, Thermococcus hydrothermalis here and Thermococcus bauophilus. And well, obviously, the genes that are responsible for the biosynthesis of these compounds remain unknown in Archaea. So when we analyzed these apolar lipids in thermococcales, we could observe a large diversity of compounds, as you can see in this chromatogram of Thermococcus bauophilus. We have compounds with C30 with 30 carbons in blue, 35 in yellow here, and 40 in brown here. And we could also observe a large diversity of unsaturation level of these compounds, ranging from 1 in the whole structure to 1 per isoprenoid unit in each of these compounds. So we did the same analysis on our 53 strains of Thermococcales. Unfortunately, we could detect this apolar polyisoprenoid in only 17 strains that we will call positive for apolar polyisoprenoid. And among these 17 strains, we had 15 that could synthesize the 3 types of compounds that we identified. Just like core lipids, we also superimposed the lipid composition of the Thermococcales on their phylogenetic tree, as you can see here. And what we could observe is that 12 of our positive for apolar polyisoprenoid strains were gathered in 3 different groups here that were closely related. And the 5 remaining strains were actually quite randomly dispersed in the phylogenetic tree of Archaea, of Thermococcales. We could invoke 2 hypotheses to explain such a distribution of apolar polyisoprenoid in Thermococcales. The first one is that the fact that this randomly dispersed Thermococcales could synthesize apolar polyisoprenoid suggests that they could have been the subject of isolated event of horizontal gene transfer and thus acquired the ability to synthesize this compound. On the other hand, we could also suggest that the species that were negative for apolar polyisoprenoid could have randomly lost the ability to synthesize this compound. But in any case, this should result in differences in the gene content of this species. For instance, we should observe genes that would be specifically present in positive strains and absent from the negative strains. So we looked at the genomes of these 53 strains of Thermococcales, and unfortunately, we couldn't identify any such genes. So we came up with other hypotheses to explain the distribution of these compounds in Thermococcales. The first one is that maybe some of the negative strains could possess the genes to synthesize these compounds, but don't express it or just at too low levels for us to detect this compound. And on the other hand, it could also come from the fact that they could synthesize this compound, but under other conditions that we didn't test here. So altogether, this suggests that apolar polyisoprenoid could actually be more widespread in Thermococcales that we could observe here. Putting all our results together that we obtained so far, what we can say is that Thermococcales share a common set of polar head groups that is well, biased. They also are all capable of synthesizing both dieters and tetraeters. And finally, the apolar polyisoprenoid could be widespread in this archaeal order. So altogether, this suggests that the membrane organization that we have proposed for Thermococcus borophilus could be valid in other Thermococcales, if not all of them. So if it's present in all of these strains, it also suggests that maybe it could be present outside Thermococcales. So this is what we will look at right now. So here you have a phylogenetic tree of all the Archaea that have been isolated so far, 500 of them approximately. For instance, you can see the Thermococcales here. And well, we didn't reanalyze these 500 Archaea strains, but we look at their lipid composition that have been described in the literature. We could come up with 440 core lipid compositions, but very often we did this characterization either lack quantification of the lipids, or they were quite biased because of the extraction procedure they used and may have missed these tetraether lipids, or some of them didn't even look at the diether composition of the Archaea strains. So because of this, we had a very inhomogeneous quality of the lipid dataset, and we tried to extrapolate the lipid composition of a closely related strain in order to infer the lipid composition of these strains that could be which composition could be biased. For instance, if a strain was described as producing only dieters, but a close relative was able to synthesize tetraeters, then we considered that our species of interest was also capable of synthesizing these tetraeters. So this ended up on 3 different groups of core lipid composition in Archaea. In yellow, we have those that synthesize elevated proportion of both dieters and tetraeters. In purple, we have those that synthesize a vast majority of tetraeters. And finally, in green, we have the species that produce only dieters. We superimpose this lipid composition on the phylogenetic tree of Archaea. And what we can see at first is that with the exception of this green group here marked by the cross, all Archaea were able to synthesize both dieters and tetraeters, obviously in different concentration proportions, but still they could synthesize both of them. And this suggests, just like for Thermococcales, that maybe the membrane organization that we have suggested for Thermococcus borophilus could be widespread in Archaea. If we look more closely, we can see that all the basal clades of this tree, for instance here, but the Thermococcales as well, or here, are in yellow, so could synthesize both dieters and tetraeters. And like for Thermococcales, this suggests that the membrane organization could be an ancestral feature of Archaea. We could also correlate the lipid composition with the optimal growth temperature on the left here, pH in the center, and salinity on the right of our Archaeal strains. And what we could observe first is that the green strains tend to live in moderate temperature with neutral pH, but high salinity environment. So they are mostly allophilic strains. The yellow strains tend to live more in high temperature settings or moderate temperature, but with neutral pH and marine salinity. And finally, the purple strains tend to live at high temperature, either neutral or low pH, and in freshwater settings. So they are mostly thermoacidophilic strains. We did the same analysis for our apolar polyisoprenoids. And as you can see here, the data was much sparser than for core lipids, as we could only come with 37 such compositions before analyzing our Thermococcales strains, of course. So we didn't extrapolate at all the lipid composition of for apolar polyisoprenoid, but we just reported the longest compound each strain could synthesize. For instance, in dark green here, you have the strains that can synthesize up to C20 compounds, while in brown here, you have those that can synthesize up to C40 compounds. We superimpose this composition on the phylogenetic tree of Archaea. And if you look at the species in black, which are those that cannot synthesize apolar polyisoprenoids, you can see that there are only few of them that are quite dispersed among the tree of Archaea. This suggests that when we looked for apolar polyisoprenoid in Archaea, we almost always found them, and thus that they are quite widespread in Archaea. So again, this suggests that inserting this apolar polyisoprenoid in that the membrane organization that we suggested for Thermococcales could again be widespread in Archaea. And similarly to core lipids, we also found apolar polyisoprenoid in the most basal clades of Archaea, and thus that the capacity to synthesize these compounds is an ancestral state in Archaea. Eventually, we also correlated this composition with the optimal growth conditions of our Archaeal strains. First, what we can see here for the temperature is that the species that live at higher temperature tend to have longer apolar polyisoprenoids. For the pH, it's the reverse trend. So species living at lower pH tend to have lower, smaller apolar lipids. And finally, those living in high salinity tend to have longer compounds as well. So this suggests that apolar polyisoprenoid could be important for the adaptation of this organism, but it also makes sense if we go back to the core lipid composition of these strains. For instance, if we look at the thermoacidophilic strains here, they synthesize a vast majority of tetraeters, and their membrane is thus mostly in the form of a monolayer, in which inserted long apolar polyisoprenoid is hard to imagine. And so that could be why they tend to synthesize smaller compounds. So putting all our data together, what we could elaborate a new scenario for the evolution of the membrane architecture in Archaea. So as I said, the membrane organization as suggested for Thermococcus bauophilus seemed to be quite ancestral state of the archaeal membrane. But it has then been conserved in neutrophilic organisms here, which synthesize both bilayer and monolayer lipids with apolar polyisoprenoids, but shifted to a completely bilayered membrane in allophilic strains, which does not preclude the possibility to form different bilayer domains, for instance, by either inserting or not apolar polyisoprenoids, and to almost completely monolayered membrane in thermoacidophilic strains. But again, this does not preclude the formation of membrane domains. For instance, the low proportion of dieters could suggest that they could constitute some kind of islands of bilayers in the monolayer C of the membrane of thermoacidophilic that could allow the insertion of specific proteins. So to conclude this presentation, I just would like to discuss some of the future directions and outlooks of my work. So first, of course, this work heavily rely on the demonstration of the novel membrane organization that we suggested for Thermococcus bauophilus. But as I said in the introduction, this remains impossible with our current methodologies. But instead, what we could gain valuable insight from is the elucidation of the physicochemical properties of the compounds that constitute this membrane organization. For instance, we know that dieters alone or tetraeters alone are able to form membrane domains, but we don't know how a mixed membrane behave. Similarly, we have only data on how apolar polyisoprenoids insert and affect bilayer membranes, and how and if they could insert in other monolayer or mixed membrane remain again uncharacterized. Eventually, we only have limited data on archaeal intact polar lipids. We know how, for instance, phosphoethanolamine dieters and tetraeters bearing phosphoglycosidic head groups behave, but the physicochemical properties of the diversity of polar head groups and of course structures that we identified remains uncharacterized as well. All these characterizations have been notably hindered by the fact that we have a poor understanding of the lipid diversity in Archaea. So I suggested to improve the characterization of the membrane composition, starting with intact polar lipids. As I said during the presentation, analyzing and extracting these lipids remain highly biased, and we have different points that we could try to improve in order to enhance the recovery of these intact polar lipids in Archaea. The core lipids in Archaea have been widely studied, but as I said again, some of these studies were biased, and re-evaluating some key species of Archaea using updated protocols could give us could actually clear the distribution of these compounds in Archaea. We also started to improve the characterization of apolar polyisoprenoids in Archaea, but the data remain very limited. And so analyzing these very interesting compounds in a more systematic way could give us some information on their adaptive function or biosynthetic pathways in Archaea. My work mainly focused on the lipid part of the membrane, as it is the foundation of the leaflet, but also of the impermeability of the membrane. But we know, especially for Archaea, that the membrane proteins can actually represent a significant part of the membrane in this organism. And again, very limited knowledge. We have a limited knowledge of the identity and functions of these membrane proteins. Last but not least, we know we have some data on how Archaea are able to synthesize diether lipids and different polar groups such as phosphoinositol. But how they synthesize all the other compounds that we identified remain largely unknown. And trying to resolve these biosynthetic pathways could actually give us means to assess the distribution of these lipids in Archaea in a more reliable way than only studying their lipid composition. And with this, I would like to thank you all for your attention, but also express my gratitude to all the fantastic people who contributed to this work, starting with, of course, my team, the M2E, but also the whole MAP laboratory, and of course, all the people who well, got their hand dirty with Archaea lipids, the molecular biogeochemistry group of Pierre Adam and Philippe Schaeffer in Strasbourg, the molecular organic geochemistry group of Vincent Grossi in Lyon, and also the organic geochemistry group of Kai-Uwe Inrich in Bremen. And finally, all the people who provided invaluable scientific, financial, and psychological support. Thank you all.